Hi, welcome to my presentation on viral genomics, diversity, and ecology. So first we'll talk about how viruses are classified. It's actually been really difficult to classify viruses into a standard taxonomy because it's really difficult to suss out what their evolutionary relationships are. They have all different formats in their genome. Some of them are DNA, some of them are RNA, some of them have double strands, some of them have just one strand. Um, they've probably evolved at multiple points and very little is known about their evolution. And then just the simple fact that their genome is so tiny, they'll only have a few genes in there. And that makes it really hard to figure out, the evolutionary relationships between different genes. A lot of them will have completely distinct genes from each other. And, if they don't have kind of genes in common, then you can't really analyze the sequence of their genes to figure out anything about their evolutionary relationships. And then finally, you have so much horizontal gene transfer going on with viruses. It's not uncommon for them to accidentally pick up pieces of their host genome and incorporate that into new viral particles. So that means that when you're looking at a viral genome, you can't just assume that all of the information in there is actually coming from that virus or from that virus's ancestors. Some of it may have come from a host that was infected by the virus at one point. So instead of using kind of a typical taxonomy for viruses, the most common way to classify viruses is using the Baltimore scheme, which was developed by this guy, David Baltimore, who was kind of an early virologist. He actually won the Nobel Prize along with two of his lab mates for discovering the reverse transcriptase enzyme that retroviruses have. That enzyme has been really extremely useful in the lab. It's really kind of revolutionized the way that we're able to work with DNA and RNA in the lab, as well as just kind of overturning the central dogma of biology a bit, right? The central dogma states that information is stored in DNA, it's transcribed into RNA, and then that's translated into protein, and then protein does work. Reverse transcription goes against that scheme. In reverse transcription, you have information going from RNA back to DNA. So before we knew about reverse transcription, no one believed that that was possible. So it was really a significant find to figure out how viruses are actually doing that using the reverse transcriptase enzyme. Under the Baltimore scheme, there's seven different groups or 7 different classes of viruses. They're classified based on 3 things. First, the format of their genome. Like is it DNA, RNA, single-stranded, double-stranded, plus sense, minus sense. Then how are they getting their genes into mRNA format so that they can be translated by the host ribosome. No matter what genome the virus starts with, it has to be able to produce something that looks like mRNA so that the host ribosomes can translate it. Otherwise, it's impossible for them to have their viral proteins made. And mRNA is always plus sense single-stranded RNA, so they have to make something that looks like that using their genome. And they have different ways of getting that accomplished. And then finally, the Baltimore scheme is based on how viruses are replicating their genomes, how they are making extra copies of their genomes. So different viruses will have different strategies for that as well. And before we continue, just a reminder about plus sense and minus sense. Plus sense means the sequence of bases in the strand is the same as for mRNA, so that it basically has the correct codons in it, and you can feed that information into a ribosome and it will be able to produce a protein that is the correct sequence of amino acids. So our mRNA is plus sense, of course. You also have in DNA the template strand that's used to make the RNA. The template strand is minus sense. It is complementary to the mRNA, but is not the same as the mRNA. It doesn't have the same information. So in a minus sense strand, you don't have the right codons that you would need to have the ribosome read it and make protein based on it. If you tried to make protein based on a minus sense strand, you would not get the correct protein. Every amino acid would be wrong because all the codons are wrong in the minus sense strand. But they are complementary to the correct codons in the right strand, which is plus sense, or complementary to the right codons in the plus sense strand. So you can use a minus sense strand to build a plus sense strand as a template. So in double-stranded DNA, the template strand is the one that is used to build mRNA. It is complementary to the mRNA, so it is minus sense. The mRNA itself is plus sense, and the other strand of DNA, the coding strand, is plus sense as well, because the sequence of bases in the coding strand is the same as the sequence in mRNA. The only difference is that instead of U's, you have T's in the coding strand. So if you look at the sequences of those strands, here's your RNA, your mRNA, so you have AUG, AUC. So this is our plus sense strand. The coding strand is the same sequence. Instead of AUG, it's ATG. Instead of AUC, ATC. So instead of having U's, you have T's. But apart from that, the sequence is the same. So if you were to use this to try to build a protein, you would be able to do it. The information in there is correct. The only thing you would need to do is replace those T's with U's, because a ribosome can only read RNA, not DNA. Then the template strand, which is minus sense, does not have the same sequence of bases as the mRNA has. Instead of AUG, it has TAC. Instead of AUC, it has TAG. So that's completely different. If you were to feed the template strand into the ribosome and try to get a protein, assuming this ribosome was able to work with the DNA as a template, you would not get the correct protein at all. All the codons would be wrong. So the first two groups of viruses in the Baltimore scheme are DNA viruses. They're called that because their genome is DNA, not RNA. So group 1, or class 1, in the Baltimore scheme is viruses that have double-stranded DNA genomes, which is kind of like a normal genome. It's basically the same format as the genome of any cell. So since the genome is already in the format that cells use for their genome, it's pretty simple for a group 1 virus to replicate their genome and to get it transcribed into mRNA. The host polymerases are already able to work with their genome because it's double-stranded DNA, or dsDNA. So host polymerases will make more copies of the genome by replication, and they will make mRNA based on the genome by transcription, and then that mRNA is used to build viral proteins. Group 2 viruses in the Baltimore scheme are single-stranded DNA viruses, or ssDNA. Technically, they can be plus sense or minus sense, but virtually all of them are plus sense. It's very, very rare to have a minus sense single-stranded DNA virus. And that's probably the main reason why the minus sense single-stranded DNA viruses don't have their own group in the Baltimore scheme, just because they're so rare. So with these single-stranded DNA viruses, most of them plus sense, when they infect a host cell, first they're going to make a complementary DNA strand using their single-stranded DNA genome to build, another strand of DNA, and that gives them double-stranded DNA. So now they have something that looks just like a regular genome to the host cell. So now host cell polymerases are able to replicate that genome and transcribe it to make mRNA from it. So here you have kind of a diagrammatic summary of that. The group 1 viruses are double-stranded DNA. So here's their double-stranded DNA genome. The host cell polymerases are already able to work with this to replicate it and to transcribe it into mRNA. Here you have your class 2 single-stranded DNA viruses. Most of them are plus sense, so their genome would have just the one strand. So the first thing they need to do is synthesize the other strand, which would give them a two-stranded DNA genome, basically. It's not properly their genome because their genome isn't normally in that format. It's kind of an intermediate. And then using that double-stranded DNA intermediate, the host polymerases are able to replicate it and, and make mRNA based on it. Groups 3, 4, and 5 in the Baltimore scheme are the RNA viruses, which means that they have RNA as their nucleic acid in their genome. They do not have DNA. So since they have RNA genomes, their replication is going to be a bit more complicated than it is for the DNA viruses, just because their genome is in a very different format from the genome of the host cell. So in order to replicate themselves, they need to make more copies of their genome. They need to replicate their genome. But the host cell actually doesn't have any RNA polymerases that can make RNA using an RNA template. The host cell polymerases are designed only to use a DNA template, and therefore transcription. So the host cell RNA polymerases use a DNA template to build RNA. But these viruses need to use an RNA template to build more RNA, since their genome starts as RNA. That means they need to have a special enzyme, which is RNA replicase. RNA replicase is an RNA polymerase that uses RNA as its template instead of DNA. So once one of these viruses gets into the host cell, the RNA replicase enzyme the RNA replicase enzyme is going to be inside the capsid of the virus. Once the virus gets into the host cell or injects its contents into the host cell, the RNA replicase enzyme will be activated, and it will use energy that comes from the host cell to build, and nucleotides that come from the host cell to build new copies of the viral genome in the RNA format that genome is in. So for group 3, you have double-stranded RNA viruses in that group, or dsRNA. So their genome is RNA and it has 2 strands. One of those strands is already in the format of mRNA. It would be the +sense strand. So the RNA replicase that the virus has is both going to replicate the genome and it's going to make extra copies of that +sense strand, which will serve as mRNA for the virus. In group 4, you have +sense single-stranded RNA viruses. So their genome is already +sense single-stranded RNA, that is mRNA format. So their genome can directly be used as mRNA to build viral proteins. They also have the viral RNA replicase enzyme to make more copies of their genome. Then you have the group 5 viruses that are -sense single-stranded RNA viruses. So their genome is not in mRNA format, but since it's -sense, it can be used as a template to build something +sense that would be in mRNA format. So the viral RNA replicase for these guys is going to build +sense strands based on the original genome, using the original genome as a template to build these +sense single-stranded RNA bits, which are serving as the mRNA for that virus. Then the viral replicase is also going to use some of those +sense strands that it made as a template to make more -sense strands to replicate the genome for the virus. So you have a diagram here showing those groups of viruses. First, group 3, the double-stranded RNA viruses. One of their strands is already in mRNA format. The RNA replicase is going to replicate the genome, making more copies of it. And it's also going to make extra copies of that +sense strand, using the minus strand as a template to build more of the +sense strand, which serves as the mRNA. In the class 4 viruses, those are +sense single-stranded RNA, so their genome is already in mRNA format, they just use it for mRNA. You also have the RNA replicase that's going to make more copies of their +sense single-stranded genome. Then in class 5, you have the -sense single-stranded RNA viruses. So their genome is not useful for mRNA, but it can be used as a template to build mRNA or +sense, single-stranded RNA. So the viral RNA replicase is going to use their -sense genome as a template to build plus sense strands that serve as mRNA. And it's also going to replicate the genome. And here you have a graphical comparison of those RNA viruses. Starting with class 3, the double-stranded RNA viruses. That one is actually on the far side. So you start with your double-stranded RNA genome. The two strands in the genome would actually separate so that each of them can be used as a template to build the other strand or to build extra copies of the other strand. So your RNA replicase is going to start using the minus sense strand and the plus sense strand as a template to build the other strand, which gives you more copies of that double-stranded genome. Also, the RNA replicase is going to end up making extra copies of this plus sense strand that will serve as mRNA for translation of the proteins of the virus. Then class 4 or group 4 is your positive sense or plus sense ssRNA viruses, and that's far over here on the left. So when that type of virus infects a host cell, its genome is just plus sense single-stranded RNA. So that's going to serve directly as mRNA for translation of the viral proteins. But also the RNA replicase is going to use that as a template to build a minus sense strand. Then it can use that minus sense strand as a template to make more copies of the plus sense strand. So that's how the viral replicase would be making more copies of that genome. Then group 5 is the minus sense RNA, single-stranded RNA viruses. So here's that minus sense single-stranded genome. This does not work for mRNA. It can be used as a template to build mRNA, but it is not in the format of mRNA. So you have the RNA replicase using this as a template to build a plus sense strand. That plus sense strand can serve as mRNA for translation of the viral proteins. And the viral RNA replicase is also going to use it as a template to build more copies of the minus sense strand, and that's how it's replicating that genome. Then the last two groups in the Baltimore scheme are kind of special. First, group 6 is the retroviruses, which are abbreviated, well, they have a plus sense single-stranded RNA genome, and they're abbreviated plus ssRNA-RT, the RT for reverse transcription. And then they're just commonly called the retroviruses. So these all have a plus sense single-stranded RNA genome, which means that it is already in mRNA format, but instead of just using it as mRNA, they're going to do something much more complicated and use their reverse transcriptase enzyme to reverse transcribe that RNA genome to DNA. They're actually going to make double-stranded DNA intermediate genomes. And most of them will then insert that double-stranded DNA copy of their genome into the host chromosomes using the enzyme integrase. Then, once it's in the host genome, it's going to remain dormant for some period of time, possibly short, possibly long. At some point, the genome will reactivate, and the host cell polymerases then will transcribe it into mRNA that will then be translated into viral proteins, and the host polymerases will also be replicating the double-stranded DNA in a genome intermediate. So here you have your retroviruses in this diagram. They start with a plus sense single-stranded RNA genome. They use reverse transcriptase to reverse transcribe that genome to DNA, creating a double-stranded DNA copy of the genome. They will usually insert that genome into the host chromosome using the integrase enzyme. Then later on, when it reactivates, the host cell polymerases will transcribe this into mRNA, which would be used in translation to build viral proteins. Finally, the last Baltimore group is the DNA retroviruses, or the dsDNA-RT viruses. So these viruses have a double-stranded DNA genome, and they also use reverse transcription, even though their genome already starts out as DNA. So since they have a double-stranded DNA genome, you'd think it could be simple. The host cell polymerases could just replicate it and transcribe it, but instead they're doing something very complicated. So first, their genome is not actually completely double-stranded. You have one strand that's complete, and then the other strand is just like a partial strand, so it doesn't cover the entire genome, basically. So the first thing that's going to happen when this type of virus infects a cell is that a viral polymerase is going to complete the second strand, so that they actually have a complete double-stranded DNA genome. Then the host cell polymerases will use that double-stranded DNA genome as a template to make mRNA and to make something called the RNA pregenome, basically copying the entire genome into RNA and then calling it a pregenome. Then the reverse transcriptase enzyme comes into play. It will use that RNA pregenome as a template to reverse transcribe a DNA genome based on it, and that is the way that they copy their genome. So this diagram here is showing the genome of a typical dsDNA-RT virus along with the transcripts, the RNA transcripts that are made based on that genome. And then this is showing kind of the, I guess, the sequence of nucleic acid formats that the information in their genome goes through. So starting up here with the genome diagram. In green, you have DNA. In yellow, you have RNA. So their original genome is in the center here. You have one complete strand in dark green and one incomplete strand in light green. So this is their incomplete double-stranded DNA genome. The first thing that will happen is the viral polymerase is going to start adding to this incomplete strand until it goes all the way around and completes the circle. So at that point, they have a completely double-stranded DNA genome. Then the host cell polymerases will use this genome as a template to transcribe all of this yellow RNA based on it. The short pieces of RNA here are the mRNA. That's going to be used in translation to make viral proteins. And then this long piece of RNA that's actually the entire length of the genome, that is the RNA pregenome. So all of the information in the genome is contained in this RNA pregenome. Then the reverse transcriptase from the virus is going to use this RNA pregenome as a template to build another copy of the DNA genome, the original DNA genome. And then this is kind of summarizing that. So you start with your double-stranded DNA genome, then you have your viral polymerase complete it so that it's actually completely double-stranded. Then you have your host cell polymerases build mRNA using that genome, so they will transcribe it to mRNA. And they also build the RNA pregenome, which is shown right here. It's a plus sense RNA strand. Then you have the reverse transcriptase enzyme use this pregenome as a template to build a DNA strand based on it, and then that DNA strand is used as a template to build the other DNA strand, and that gives you back your original dsDNA genome, which they will make in an incomplete state and package into new viral particles. There's not a lot of viruses that do this. The DNA retroviruses are pretty uncommon, but you know, some of them are managing to do it this way. If you look at the Baltimore group of different viruses and the hosts for those viruses, you find that not every Baltimore group will infect every type of host. So for archaea, we only know viruses in groups 1 and 2 that can infect archaea, which are the DNA viruses. For bacteria, we know viruses in groups 1, 2, 3, and 4 that can infect bacteria. But actually most of them are in group 1, so they're double-stranded DNA viruses. For protists, we know viruses in groups 1 and 3 that can infect them. For plants, every group of virus can infect plants except for group 6, the retroviruses. So everything except for the retroviruses can infect a plant. For animals, every group of viruses can infect animal cells. Then for fungi, it's only groups 3 and 4 that are known to infect fungi. If you're comparing prokaryotes to eukaryotes, you find that group 1 is the biggest group of viruses that infect prokaryotic cells. So that would be the double-stranded DNA viruses are the most common type of virus to find infecting a prokaryote. Then for eukaryotes, group 4 is the biggest, which is the single-stranded plus sense RNA viruses. So most viruses that, well, maybe not most, but the single biggest group of viruses that infects eukaryotic cells would be those plus sense single-stranded RNA viruses. Then the graph is just kind of breaking that down. So it's showing first for the different domains of life, archaea, bacteria, and eukarya, the types of viruses that infect them. And then over here it's showing for the different types of eukaryotes, the types of viruses that infect them and kind of how common they are, or how many viruses from each group are known to infect that type of host. So for plants here, you have 18, 348, 40, 500, 1, and 55. So you have 18 viruses in group 1 that can infect plants or that are known to infect plants, 348 viruses in group 2 that are known to infect plants, 40 in group 3, 500 in group 5, in group 4, 1 in group 5, and 55 in group 7. None in group 6. So some viruses can actually cause cancer instead of just killing the host cell. Cancer is any time that you have cells in the body that are dividing in an uncontrollable fashion. So normally cells in the body are very tightly regulated as far as when they are actively dividing. And most of them are not actively dividing at any given time at all. If you had cells that were just kind of dividing all over the place whenever they wanted to, then you would end up kind of losing the structure of your organs and the different parts of your body. And when your organs lose their structure, then they also lose their function because the function is dependent on the structure. So it's important for you to be able to prevent your cells from just going through cell division. And there's a lot of kind of safeguards that cells have to prevent them from going through the cell cycle and dividing at an inappropriate time or when they're not supposed to. In cancer, you kind of lose those safeguards so that the cells will just start dividing. As soon as they're able to divide, they divide again. And then they start building a tumor. That tumor, as it grows, it can disrupt the function of the cells around it because it's just, kind of squeezing them out of the way and putting pressure on them. But also, as the tumor develops, you're going to have a lot of mutations going on in those cancer cells because they're just kind of rushing through the cell cycle. They don't have an opportunity to repair their DNA. Normally, DNA is repaired in between cell divisions. So if you're just going from one cell division to the next, you don't have time to repair your DNA. So any mistakes that get made when you copy the genome for cell division, they're going to stay as mutations. Because of all these frequent mutations, cancer cells are going to be kind of unlocking genes that are normally kept silent, normally inactive, and not transcribed. So every cell in your body, of course, has the genes, has all of the genes that you have. All of your cells have the same genes. So a skin cell is very different from a neuron, but they both have the same genes. The way that those cells are made to look and behave different is by controlling which genes are transcribed at any given time. So that's very tightly regulated in cells. Certain genes are never to be used by cells of certain types. And so you'll have those cells producing a protein, usually that prevents, or some type of protein that prevents those genes that they're not supposed to use from being transcribed. In cancer, you can get mutations to the genes that are used to build those proteins that prevent gene transcription that's not supposed to occur. So once you have mutations to those genes, maybe you start making proteins that don't work. So now those proteins are not able to prevent transcription of genes that this type of cell should not be transcribing. Now you're going to have transcription of those genes. So in this way, cancer cells can gain abilities that they didn't start out with. They can gain the ability to dissolve the extracellular matrix that they're in. They can gain the ability to cause new blood vessels to grow towards them. And these types of abilities help the tumor get very, very large and help cancer cells move out of the tumor into nearby body tissues. And at that point, things get very, very serious because the cancer cells can move to a new location and start a new tumor there. So cancer is real serious, as you know. It takes a lot of mutations to cause cancer. You need to have unregulated cell division, and cell division is very tightly regulated. The cell has multiple mechanisms to prevent cells from dividing when they shouldn't. So you have to have mutations that defeat a lot of those mechanisms to actually cause uncontrolled cell division. Also, every cell has the ability to sense when it's taking damage, and if it becomes kind of too damaged, the cell should enter apoptosis and basically destroy itself to prevent damage to neighboring cells. In cancer, not only do you need to have mutations that increase cell division, you also need mutations that prevent apoptosis, that prevent the cell from being able to enter apoptosis when the time is right. So now you have a cell that can't stop dividing and can't kill itself. That's cancer. Then for the cancer to be extra serious, you need additional mutations that allow those cells to invade other tissues. So there's a lot of things that can cause cancer. Essentially anything that damages your DNA has the potential to cause cancer. But you have some viruses that actually either sometimes lead to cancer or kind of reliably lead to cancer. Those viruses include HPV, the Epstein-Barr virus, hepatitis B, hepatitis C, and HIV, and others. Out of those, the most common one is HPV, the human papillomavirus, which causes genital warts, but it can also cause cancer. It's most dangerous for women. It causes cervical cancer. It's actually the leading cause of cervical cancer, but it can cause cancer in men as well. It's just not as common. So HPV is a very common sexually transmitted virus, and there is actually a vaccine for it. So if you have not been vaccinated for HPV, you should probably do that. All these viruses that are causing cancer typically fall into a couple different groups. They're either the DNA viruses in groups 1 and 2 in the Baltimore scheme, or they are the retroviruses in group 6. And there's reasons why these viruses are the ones that typically lead to cancer or could lead to cancer. For the DNA viruses, they usually don't have kind of special enzymes that are used to replicate their genome. Instead, they're relying on the host to replicate their genome. So the host has DNA polymerases, that'll use DNA as a template to build more DNA that can copy the viral genome. But those host polymerases aren't active all the time. The host is only going to copy its genome if it's preparing to divide, which means that normally you don't have those DNA polymerases in the host cell that can copy DNA. You just don't have them. You're not going to make them unless it's time to actually copy the DNA for cell division. So that means it is to the advantage of these DNA viruses to kind of force the host cell to enter the cell cycle and start dividing. They are only able to be replicated in a host cell that is actively proceeding through the cell cycle, because that's the only type of host cell that is going to make the DNA polymerase that they have to replicate their genome. So that's the reason why a virus like HPV will cause genital warts. It is actually causing its host cells to enter the cell cycle and start dividing, and that creates a wart. This little accumulation of extra cells that are all infected with that virus. It's the wart. So So it's kind of a testament to the regulation of the cell cycle that even that is not enough to cause cancer most of the time. Usually if you have HPV, you just get the warts, but sometimes it can progress to cancer, cervical cancer, or a different type of cancer. Then for the retroviruses, they can also cause cancer, but by a different mechanism. For the retroviruses, usually they're going to insert their DNA genome copy into the host cell chromosome using the integrase enzyme. And there's not really any way for the virus to control where it's going to insert. It's just kind of at random for the most part. So that means that it's possible that this viral genome could insert right in the middle of a gene. And if that happens, that gene is basically going to be lost. And, it's damaged. There's no way to make protein off of that gene anymore with the viral progenome inside of it. If that gene that the virus inserted in the middle of is, supposed to be making a protein that controls cell cycle progression or allows the cell to enter apoptosis, then having that retrovirus there makes it more likely for the cell to become cancerous. So we'll talk a little bit about the probable evolution of viruses. Not very much is known about this topic because it's really hard to investigate because viruses don't have rRNA. So for all life forms, you can use the rRNA sequences in their genome to kind of analyze the probable evolutionary relationships between those organisms. Since viruses don't have ribosomes, they don't have ribosomal RNA, it's impossible to do that for viruses. They have very few genes at all. So when scientists are trying to investigate the evolutionary history of viruses, instead of relying on genetics, they use proteomics. So just like the genome is all of the genetic material contained in a cell, the proteome is all of the proteins that are contained in a cell, or of course in a virus. And proteomics is the study of the proteome of different cells or viruses. So you can get a lot of information from proteins and protein structure about the evolutionary relationships between proteins, and then you can extrapolate from that to get information about the evolutionary relationships between organisms that have these proteins. Of course, you have the primary sequence of the proteins, or the primary structure, the sequence of amino acids that they have. That tells you some information about the genes that gave rise to these proteins, and it tells you a lot about how different proteins are related to each other, and you can kind of estimate how long ago two proteins that are very closely related diverged evolutionarily. But you actually can get information from the secondary structure and from the tertiary structure of proteins as well, although that's a more complicated process. So most of what we know, or most of what we think about the evolutionary history of viruses is based on proteomic information, on having studied the proteins that they have and the structure of those proteins. Based on that, we have figured that probably viruses evolved before the last universal common ancestor lived. So during the very earliest stages of life, before you had 3 domains of life, and before you had a common ancestor for all cells. So of course this would have been during the RNA world, when there was no DNA and there was only RNA. And actually it does seem that RNA viruses are older than DNA viruses, which would make sense if viruses evolved during the RNA world period. The oldest group of viruses of all seem to be the group 3 viruses, which have double-stranded RNA. The top diagram here is showing kind of a summary of a possible evolution of viruses. So you start out with some cells that are becoming kind of smaller and smaller and simpler and simpler. Their genomes are getting smaller and smaller. It was beneficial to be very small because you don't need as many resources and you can divide more quickly if you're smaller. So because of that, it's possible that some cells were under a strong selective pressure to get smaller and smaller. And eventually they would have lost basically the structures of the cell. They would have lost like the cell membrane, and they would have lost a lot of the genes that they needed to live independently. And at that point you would have had a virus that is not able to replicate itself without a host. So the hypothesis is that some of these early cells lost so many of their genes that they could not survive without a host anymore. At that point, they would have been a virus in a protein capsid that needs to, find a host cell and exploit the resources and machinery of that host cell to create new viral particles. So it's possible that this process went on before LUCA even existed. It's also possible that viruses played potentially a really important role in the evolution of cellular life and in the conversion of RNA to DNA as the genome, as the molecule of the genome of all cells. So in this kind of idea of how viruses might have played a role in cellular evolution, first, we think that viruses may have evolved the reverse transcriptase enzyme. Then they could have used that enzyme to create a DNA genome using an RNA template. So these viruses would have been RNA viruses with RNA genomes. They might have used this enzyme to create DNA genomes. The advantage of having a DNA genome in an RNA world is that host cell defenses would be, at this point, all based on the assumption that the virus will have an RNA genome. If the virus has a DNA genome instead, it would be possible for the virus to avoid all of the defenses that the host cell might have. But actually, in order for a virus to get a DNA genome using reverse transcriptase, it would also need some other enzymes as well, because it would need to build DNA nucleotides. So it would need to convert RNA nucleotides into DNA nucleotides, and then have the reverse transcriptase enzyme to allow reverse transcription of their RNA genome to DNA. So that's a possible origin of DNA viruses. Once you have DNA viruses that are using DNA genomes, those viruses would have needed to evolve DNA polymerases and other enzymes that are needed to work with DNA so that they would be able to replicate their DNA genomes. Then, if you had one of those DNA viruses infect a host cell and transfer some of those genes for the DNA polymerase and stuff like that to the host cell by horizontal gene transfer, then the host cell would gain some of these tools that it would need to make its own DNA genome. And then the host cell could have used those tools to actually convert its genome to DNA. If you had a host cell that did this, that used viral proteins to convert its genome to DNA from RNA, that host cell would actually have an advantage, assuming that it survives the viral infection, of course. Because DNA is more stable than RNA. Actually, if you have RNA in water and you just wait, it'll spontaneously degrade by reacting with the water. DNA does not do that in water. So DNA is just kind of an inherently more stable molecule, and that means it's better for storing information than RNA is. So once you have a DNA genome emerge in an early cell, that cell will have a significant advantage over other cells and would have come to dominate the population. So in this hypothesis, after you have some early host cell get a DNA genome from a virus, that host cell would have gone on to be the ancestor of LUCA. And then once you have LUCA, after that you get the 3 domains of life that we know today. So this diagram is kind of summarizing that whole spiel. On the top you have some of the enzymes that you would need to actually create DNA nucleotides from RNA nucleotides. So ribonucleotide reductase, then thymidine synthase to replace the U's from RNA with T's for DNA. At that point, if you also had reverse transcriptase, a virus would have been able to create a DNA genome based on its RNA genome, and at that point you would have a DNA virus. Actually, to be a proper DNA virus, it would need to have also evolved some DNA polymerases. So once it evolved some DNA polymerases, then you have a proper DNA virus. Then if this virus infected a host cell that had an RNA genome, because we're still in the RNA world, possibly this host cell could have gotten some genes from the DNA virus by horizontal gene transfer, genes for DNA polymerases, genes for creating DNA nucleotides based on RNA nucleotides. And at that point, it would be possible for this host cell to become a DNA-based cell that is using DNA for its genome instead of RNA, assuming that it got those genes from the virus and assuming that it survived its infection with the virus. Then this cell would have gone on to create the DNA world, eventually replacing all cells that have RNA genomes with cells that have DNA genomes. Then you would have LUCA and the 3 domains of life. So a little bit about the ecology of viruses, or just how they exist in natural environments. There's a lot of viruses out there. So there's a lot of cells, especially if you're talking about, prokaryotic cells. There's a lot of them, but there's a lot more viruses than there are prokaryotic and eukaryotic cells on Earth. Possibly 10 times as many viruses as there are cells. Viruses live with cells, of course, because they're dependent on cells. Actually, every type of organism has viruses that can infect it, and everywhere on Earth where you find organisms living, you'll find viruses there as well, hoping to parasitize off of them. As with bacteria and archaea, most viruses we don't know anything about. We haven't discovered them, we haven't cultured them, we know nothing about them. So most viruses probably remain to be discovered. We think that in nature, probably viruses will follow the lytic life cycle and destroy host cells more commonly if the host cells are plentiful in the environment. If host cells are not very plentiful, then we think it's more likely that viruses would follow a lysogenic life cycle, kind of infect a host cell and then remain dormant for a long time before eventually reactivating. Just because it makes sense that if there's not a lot of hosts around, it's not a good idea to kill the host that you found. If there are a lot of hosts around, then you can kill the host that you're in right now and you'll find another one very easily. So it would just make sense. And there is some experimental evidence to suggest that it may be the case. Viruses play kind of an important role in ecosystems that have them, which is all ecosystems, because when they destroy a host cell in the lytic life cycle, the contents of that host cell will be released. And then you have other microbes around there that maybe aren't able to destroy a cell on their own, but when the contents of the cell are released, they can they can feed off of all of those organic molecules that are coming out of the destroyed cell. So when viruses destroy a cell, they're actually providing food to other cells in the area that can scavenge the remains. Also, viruses have played a very important role in the evolution of microbes, and not just prokaryotes but also eukaryotes, because of horizontal gene transfer. So for prokaryotes, there's several different mechanisms for horizontal gene transfer that are possible. For eukaryotes, it's really just viruses. So when you see evidence of horizontal gene transfer having occurred in eukaryotes, that almost certainly means that it was a virus that kind of did that horizontal gene transfer, that transferred genes between one host and another. When you see evidence of horizontal gene transfer in prokaryotes, you can't know for sure a lot of the time, like what was actually the vehicle for the transfer. Was it a virus or was it something else? Sometimes you can know for sure, but a lot of the time you can't. We do know that horizontal gene transfer has, been very important in in evolution. And so since viruses are one of the main ways that that can occur, or one of the most common ways, then we know that viruses must have been themselves very important in the evolution of other organisms. And we actually do have some experimental evidence showing how this can occur. So there have been experiments where they've cultured cyanobacteria and infected them with bacteriophages on purpose, and then introduced those bacteriophages into new populations of prokaryotes that are not phototrophs, and they do not have any of the genes that they need for photosynthesis. And then you can see these non-phototrophic cells gaining genes that they need to build photosystem 2. So the only way they could have gotten those genes would be if the virus carried them from the cyanobacteria that it previously infected to these new hosts. Another way that viruses have impacted the evolution of their hosts is just by providing a selective pressure for host cells to evolve defenses to viruses. So every cell has actually a lot of different defenses against viruses because viruses are very common. And viruses themselves have ways around host cell defenses. They have ways to sabotage those defenses and kind of force the cell to do the virus's bidding. So host cells and viruses are kind of evolving together, each trying to defeat the other in kind of an evolutionary arms race. So every time you have a host cell that evolves a new defense against viral infection, you're going to have a new strain of virus evolve that can defeat that defense. Then you're going to have a new strain of host evolve that has a new defense or has a way to beat the virus's way to beat the host cell's original defense, and so on and so forth. It's just tit for tat, back and forth, basically forever. So a lot of the time you'll have specific strains that have specific defenses against viruses for the host cells and specific strains of viruses that have kind of countermeasures or ways of getting around specific host cell defenses. So just by knowing the species that a host cell belongs to, that doesn't necessarily tell you which viral defenses or I guess which defenses it has against which viruses. Just by knowing what species or type of virus you have, that doesn't tell you necessarily what countermeasures it will have against specific host cell defenses. It depends on the strain. This diagram is just kind of illustrating how the arms race goes. So you have a host cell here and a virus, and this little cutout is basically a receptor or a representation of a receptor on the host cell. And then the virus's shape is meant to indicate that this virus has a surface protein that will bind to this receptor and fit it like a lock and a key. So this virus is able to get inside of this host cell. So something the host cell might do I guess as a defense against this virus is to evolve a new receptor that has a new shape. So now if the original virus tries to infect this host cell, its surface proteins do not fit the receptor anymore and it can't do it. So this host cell is safe against the original virus. But the virus is going to respond by evolving to get new surface proteins that actually do fit that new host cell receptor so that now the new virus can infect the new host cell. And this process is just going to repeat basically forever. So before we had talked about CRISPR, now we'll just talk about it a little bit more. CRISPR is a very common mechanism of defense for prokaryotic cells to have against viruses, and it works basically like an adaptive immune system for prokaryotes. In humans, your adaptive immune system kind of remembers all the pathogens that you've encountered in your life, and if you ever encounter those pathogens again, it's going to activate very quickly and come down hard on the pathogens, destroying them with extreme prejudice. So with CRISPR, host cells are able to remember which viruses they encountered in the past and also which viruses their ancestors or parent cells encountered in the past. And if they encounter those viruses again, they will be able to respond swiftly and destroy the new viral infection. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is quite a mouthful. This diagram is showing a bit of CRISPR. So CRISPR would be a part of a host cell genome, part of the chromosome, just a short segment of the chromosome. And in the CRISPR segments, you have these repeats, which are sequences of DNA that belong to the host cell. And then between them, you have these spacers, which are actually little chunks of viral DNA from viruses that have infected this cell in the past or that infected an ancestor of this cell in the past. So you have these clustered repeats. There's multiple of them in a cluster, so that's the clustered. They're regularly interspaced because these spacers are all the same size. So you have a repeat, and then, I don't know, 20 base pairs, another repeat, 20 base pairs, another repeat. I just pulled that number out of the air, by the way. They're short. These repeats are short. They're palindromic. They're palindromes. So if the sequence is TAC on one side of this repeat, it would be CAT on the other side of the repeat for a palindrome. And each of these repeats has the same sequence, which makes them repeats. So that's why it's called CRISPR. The name's really complicated, but it is trying to tell you something about the structure of these little parts of the host cell genome. The way the system works is that when you have a virus infect a host cell, it injects its DNA into the host cell, and you have Cas proteins in the host cell that can find this DNA. They're actually going to find any viral DNA that has this little PAM segment in it. They can bind to that, and then they're going to cut any DNA that's around it. So they're going to cut out a little chunk of the viral DNA. So here you have a Cas protein that has already cut out that little chunk of viral DNA, and then it's going to insert it into the CRISPR region of the host cell chromosome as a new spacer. Then in the future, assuming that this cell survives the infection with the virus, in the future, if this host cell encounters the same virus, the spacer is going to come in handy. The cell is actually going to be transcribing these CRISPR segments. Specifically, it transcribes the spacers, and once they're transcribed, we call them CRISPR RNA or cRRNA. So these little chunks here would be the cRRNA, little bits of RNA that were copied off of these spacers. So they have the same sequence as, well, not the same sequence, but the complementary sequence to the original viral DNA, which means that they can base pair with that DNA if they find it. Then you're going to have a Cas protein come along and bind these cRRNAs and make a complex with them. So here you have the Cas protein in complex with this particular cRRNA that came from, or was based on a sequence that came from this particular virus. So now this virus is infecting the cell again. It has injected its genome into the cell. And here's your original segment of that genome that had been cut out by the Cas protein in the past and inserted as a CRISPR spacer. Now this cRRNA has the complementary sequence to it, so it can base pair with it. And once that base pairing occurs, the Cas protein is going to chop this genome up. So because this is going to happen really quickly, as soon as the virus is beginning to infect the host cell, the virus isn't going to really be able to gain a foothold, and once its genetic material is destroyed, there's no hope for that virus left. It's basically a dead virus. There are actually some viruses that have a way around that. So you have some viruses that have proteins inside of them that they would inject into the host cell along with their genome, and then those proteins are going to find the Cas proteins and bind them and inhibit them so they can't do the cutting anymore. So for every host cell defense, there are some viruses that have countermeasures to that defense. I would not be surprised if there's also host cells that have proteins that can inhibit the viral proteins that can inhibit the Cas proteins. And probably viral proteins that could inhibit those proteins, and on and on. So earlier in the semester, we mentioned the human microbiome, which would be all of the microbes living on and in your body. But there's actually a human virome as well, which is all of the viruses in or on your body. So you don't just have viruses infecting you if you're actually like sick with a virus. You have them all the time. They're always there. Not just infecting you, but also infecting the microbes in your microbiome. So you have animal viruses in your virome, of course. That would be maybe infecting you. You also have bacteriophages, a lot of bacteriophages actually, that are infecting all the bacteria in your microbiome. You can also find plant viruses in the human virome as well. Those would be just kind of passing through. Basically, you would have them there because you ate a plant that was infected with viruses. Some of the animal viruses and bacteriophages would actually also have come in on the food that you eat. So they might be there, just for a little while on their way out. Everybody's virome is different, just like everybody's microbiome is different. The virome is not very well studied yet. We only know that it exists and its characteristics from doing genetic studies. So if you try to do like a genetic study on the human microbiome to figure out what all bacteria are in there, you will find inevitably a good amount of viral DNA or RNA, not just bacterial DNA. And this is kind of how we discovered the human virome and how we're trying to like categorize it. Actually, most of the viruses in your virome don't have RNA for their genomes. They have DNA for their genomes. I'm not quite sure why that is. A lot of them are fine to have. So some of them will like not infect your cells. Some of them will infect your cells but not make you sick. Some of them will infect the cells of your microbiome, potentially hurting those cells or not if they're lysogenic. And some of them actually can cause disease and be like not so good to have in there. In general, it's okay though. It's not really clear if the virome is like helpful for you in any way, but at least most of it is not hurtful. Part of the virome actually includes what's called HERVs, or HERVs, I guess, human endogenous retroviruses. So these would actually not be like intact viruses that you can find somewhere in your body. They're not virions. They're just bits of DNA that would be inside the chromosomes of your cells. So they're essentially part of your genome, which is why they're called endogenous, because they're part of you. So these bits of viral DNA in your genome have come from retroviruses. Not ones that infected you, but ones that infected a human very far in the past. Specifically, these retroviruses would have needed to infect the person's germline, so either the cells giving rise to eggs or the cells giving rise to sperm, and inserted its genome into those cells in a place that, wasn't too bad to have it, and just kind of got left there, and the virus maybe never reactivated, or maybe in this case it only inserted like a little part of its genome and it wasn't really enough for it to reactivate anyways. So now it's just there. It would have been inherited by all the children of that person who had the infection in their germline and all those people's children, and now essentially all humans have inherited it. So different people will have different HERVs in their genome, but everybody has some, and a lot of people would share HERVs. It's estimated that actually about 8% of your genome is viral. It is these little fragments of viral DNA that are left over from past infections of some ancestor of yours. It's not hurtful to have them there in general. There's like certain instances where there seems to be some evidence that one of them might be hurtful potentially, but nothing really conclusive. And certainly most of them are not harmful at all. They're just kind of sitting there doing nothing. So this diagram is kind of summarizing the human virome. So you have a selection of some viruses that can be found in the virome. Most of these are fine to have, but a few of them could actually cause disease, like the adenovirus here, that can potentially cause a cold, nothing so serious. The papillomavirus, that can cause warts. Some of them actually can cause cancer though, so that could potentially be serious. And then down here you have the pepper mild mottle virus. So that's just passing through. That's because you ate, a plant that was infected with this virus. Or I should say, this person ate a plant infected with that virus. If you were to zoom in on the person's gut, you might see something like this, where you have the person's epithelium, then that's covered by mucus to protect the epithelium. Then you have a bunch of bacteria suspended in that mucus, which would be part of the person's microbiome, and there you find the bacteriophages. So the bacteriophages would be specifically living any place that you have bacteria, and they'll be, killing the bacteria and releasing the contents of the bacteria. Then the other bacteria can eat that stuff, and actually you can eat it too. So we'll finish up by talking about subviral agents, which are not viruses, but they're kind of like viruses. Well, some of them, they're just simpler than viruses. They are things that replicate, that are not alive, that are simpler than viruses. And I guess I had the definition on this slide, of course. So subviral agents, some type of particle, could be protein, could be DNA or RNA, something particle-like that is simpler than a virus, so it doesn't have all of the parts that a virus has. That might mean that it has just the protein part, or it might mean that it has just the genetic material part, or it might mean that it's pretty similar to a virus, but it's lacking some genes that it would need to kind of get the job done. So in order to infect a host cell, it would need to have itself in the host cell, and it would need to have another virus, like a proper virus, in the host cell at the same time, and then it can replicate itself. So there's actually a lot of different types of subviral agents. A lot of them are derived from viruses. It's basically a virus that lost some of its genes and isn't really a full virus anymore and needs another virus to also infect the host cell with it in order to replicate itself. And you have some other things as well. So out of the whole world of subviral agents, which is surprisingly large, we're just going to talk about two, the viroids and the prions. Viroids are little bits of RNA that infect plants. So this little bit of RNA will get inside the plant and it'll just replicate itself or copy itself, and that's it. Sometimes that's okay, sometimes that can really be overwhelming for the plant and damage the plant and cause disease. Then you have prions that are just protein with no genetic material. Prions actually are not derived from viruses at all. They're not related to viruses, but they are little things that sort of replicate and that are simpler than viruses, so they are subviral agents. A prion is a protein that has misfolded in a way that it can actually cause other proteins nearby to misfold in the same way if they're of the same type. You have prions that infect vertebrates like us, and we've also found prions in fungi, but most of the prions that we've found in fungi don't infect the fungi. They're actually just kind of part of the fungi, and they're useful actually. So we'll talk about both of those in more detail. So first, viroids. Viroids are little bits of RNA. It's single-stranded RNA in a circle, and the RNA will fold up to have like a secondary structure. So just a little circle of folded RNA. We call that a genome because, I guess it's the genetic material of the viroid. It also is the viroid, but actually it's not like a normal genome, because usually we think about, genomes coding for proteins. Genomes have genes, and those have information to build proteins. But the viroid genome doesn't have genes. It doesn't code for proteins. You can't make anything with it. All you can do with it is just copy it and make a new viroid, and that's all that happens with that genome. Probably all viroids are actually related to each other. So at some point you had this little bit of RNA come about that was able to be copied by an enzyme, and as those copies were made, as time passes, eventually you get some mutations, and that forms new viroids. And that's probably where all viroids come from some common viroid ancestor. We only find viroids infecting plants. We don't find them infecting animals or any type of microbe. The reason for that has got to be that plants have RNA replicase. No other organism has the RNA replicase. Again, RNA replicase is the RNA polymerase that can use an RNA template to build more RNA. So if a viroid is going to get copied, it's a piece of RNA, it needs to end up as RNA. So you have to have some type of polymerase that can use RNA as a template to build another strand of RNA, which is the RNA replicase that plants have, and nothing else does. Since viroids aren't viruses, like they can't get into the host the way that viruses can. So viruses are going to have proteins that help them attach to the host. They're going to have some type of machinery or strategy for entering the host cell once they've attached to it. Viroids don't have that. It's just the RNA. So they kind of have to get into the host by getting lucky, almost. There has to be a break in the cell wall of the host. There's no way for RNA to get through a cell wall that's intact. So first, the plant must have some type of injury that has broken some of its cell walls. Then you need to have a viroid kind of floating around in the area that ends up just kind of floating on in through that break into the cell. Then you can have the RNA replicase enzyme, replicating that viroid to make quite a lot of viroids, and it can actually spread through the whole plant as soon as it gets into a single cell in the plant, because actually all of the cells in a plant are connected to each other by little holes in their cell walls called plasmodesmata. Having those little holes, those little plasmodesmata, helps the plants share resources with cells, with all the cells of the plant, and it helps the different cells of the plant communicate together easily without having to transport things across the cell wall. But it also means that once the viroid is in, it's in, and it can go basically anywhere in the plant. So that's kind of shown here in this diagram. This is in yellow here, or I guess orange, a little viroid. So this would actually be a circle of RNA that has folded back on itself to make this little stick with some bumps coming off of it. So that would be the secondary structure. It has already entered the plant through a break in the cell wall somewhere. And now it's able to go through these little holes, which are the plasmodesmata, to any plant cell. And it's actually able to travel in the vascular system of the plant to access other parts of the plant more quickly and get into every cell and be replicated by RNA replicase in every cell. In general, if a plant is infected with a viroid that's causing a disease for the plant, it looks like the plant is just kind of stunted and not getting enough nutrients. So for instance, here you have, like on this side, a healthy plant that is not infected with anything, and on this side, a plant that is like the same age, grown under the same conditions, but that was infected with a viroid. So it has stunted growth. Actually, you have a few different plant diseases that are pretty serious for agriculture that are caused by viroids. And that brings us to the prions. Prion is a protein that is misfolded, and it's able to make any other proteins around it that are the same type of protein misfold the same way. So The common prion protein that's relevant to people is called PRP. Literally, that just stands for prion protein. So that protein is actually doing something, in your cells. It's normal to have it. It has a function for you, and you need it. When it's in its regular folded shape, when it's folded correctly. If that protein becomes misfolded to form the prion shape, it'll make other PRP proteins that have the normal shape misfold to be the prion shape too. How is that happening is a question mark. We don't know. We don't know how it happens. We don't know why it happens, but we know that it does happen. And it's pretty serious. It can cause really serious diseases called transmissible spongiform encephalopathies, which is a lot of big words, but the transmissible part just means that you can transmit it from person to person, also from animals to people sometimes. The encephalopathy part means that it's a disease that affects the brain. And the spongiform part means that it makes the brain look like a sponge. So you're going to get a lot of these proteins building up in the brain. I should say a lot of these misfolded proteins building up in the brain if you have one of these transmissible spongiform encephalopathies. And once you get an accumulation of them that's big enough, it's going to kill cells that are around it. And as the disease progresses, you get just kind of more and more of these misfolded proteins building up to make, large deposits of them. And everywhere you have one of those large deposits, all the neurons around it will be dead, which makes little holes in the brain, which makes it look like a sponge. So in these pictures here, you can see on the left regular brain tissue, a slice of regular brain tissue, and on the right, a slice of brain tissue that is affected by one of these transmissible spongiform encephalopathies. So you have all these holes here that are not present in the healthy brain tissue. These represent places where neurons have died as a result of too many of these misfolded proteins being present. Some examples of that type of disease would be the Creutzfeldt-Jakob disease, which there's several different variants of. Also mad cow disease, which is one that you would actually catch from cows that were affected by it, and it affects cows. Some others as well. There's actually a number of them. What they all have in common, apart from chewing the brain up and making it holy and being transmissible, is that it takes a long time for the disease to develop. It takes years for these proteins that are misfolded to accumulate to levels that they're actually killing neurons. They are fatal, and there is no cure for them. So it takes a really long time for it to develop and start affecting you in a major way. But there's no way to prevent it from happening once you've got the disease, and it will just make holes that are bigger and bigger until they're too big and you die. Then this diagram here is just showing that prion protein. On this side, on the left, you have the regular version. It's called cellular PRP to reflect that it's normal, and we abbreviate that CPRP or PRPC. And then here you have the misfolded version, which is called prion PRP, which is kind of funny, prion protein, but prion P. Prion PRP, gosh. Or PPRP or PRPP. So the shape is not that much different, but functionally it's a lot different. If you have one of these proteins that is shaped like this around a bunch of proteins that are shaped like this, pretty soon they're all going to be shaped like this. Well, actually I should say it takes a few. If you have a few proteins shaped like this around a bunch shaped like this, eventually they'll all be looking like this. So prions in general are really stable, unfortunately. They're very hard to denature. Normally it's not that hard to disrupt the structure of a protein and denature it. You could use heat, you could use extreme pH, a variety of things. You could use different chemicals. But for prions, it's not really that way. They're so resistant to heat that they'll go right through an autoclave. It doesn't bother them. They'll come out the other side just as infectious as when they went in. So that means that they will last basically forever in the environment. If you are infected with one of these diseases, or if you have an animal that's infected with the disease, it's going to be kind of shedding these prion proteins in all of its excretions, the urine, the feces, the blood, all of the fluids, basically. The largest accumulation would be in the brain, though. In order to actually get rid of them, it's extremely difficult to do. You could use really, really high heat, like even higher than what's in an autoclave. So if you actually burn something, then you can actually denature the prions. But most things are kind of destroyed when you burn them, so that's not very effective practically. So what they have now is a product called prionzyme, which is an enzyme that's specially designed to actually not denature your prions, but to chew them up into little bits. So if you're kind of working in a hospital where you have patients that are known to be infected with prion diseases, you would probably be using prionzyme or a similar product on all the kind of equipment that's used to treat those people, and all of their kind of bedding and stuff like that to make sure that the prion disease doesn't spread. Prions have been known since 1982 when they were discovered by a guy called Stanley Prusiner, who won the Nobel Prize for it. The prion that he discovered was actually PRP, or prion protein, and he found out that it's causing these transmissible spongiform encephalopathies. Before that time, we really hadn't known what is causing them. Since that time, we have found more prions. We found some in fungi, particularly in yeast. And the ones we found in yeast are actually helpful for the yeast. The yeast is using them to kind of regulate its gene expression and adapt to environmental changes so that it can kind of turn on expression of certain genes when it needs them. And actually, we found a beneficial prion in humans as well, real recently, which is called MAVs. And it's kind of funny because even though it's beneficial, it still kills cells. So I guess the MAVS prion is formed in cells that are infected with a virus. And once it's formed, it causes other MAVS prions to form, and they make an accumulation, an aggregate, and then that kills the cell. And it also causes an immune response. So this is not good for the cell that is infected with the virus. That cell dies. But the cells that are around that cell are saved because the virus is not able to complete its life cycle, right? So it infects the cell, but it's not able to actually assemble and release new virions that could infect neighboring cells. So the infection is kind of shut down before it gets serious. So we'll talk a little bit more about the prion protein specifically. So you can find that protein in cells all across your body, but the highest concentration of it is in the brain. And what's weird is that no one knows what it does, but we know it does something important. We know it's important because the gene for the PRP protein is highly conserved, which means that all the mammals have it, and it always looks, pretty much similar. So it doesn't evolve much, which generally means that when you have a mutation to that gene, the animal doesn't live, or it doesn't, at least doesn't produce a lot of offspring. So those mutations are kind of eliminated from the gene pool. And yet we don't know what it does. What is that protein doing? It's doing something that matters. There's some evidence that it might be helping to transport copper into cells, but we don't know for sure. It's doing something important. You have two shapes of the PRP protein that are, that could be found in nature. First, you have the regular shape, cellular PRP. Then you have the prion shape, or prion PRP, which actually I told you guys wrong a couple slides ago. That's not abbreviated PPP, PPPPRP or whatever is abbreviated PRPSC. The SC there is coming from scrapie, because actually when this protein was first discovered, it was found in sheep, specifically sheep that have the disease scrapie, which is a transmissible spongiform encephalopathy. And then later on, we found out that it's this same prion protein that's causing, the mad cow and the Creutzfeldt-Jakob and all these others transmissible spongiform encephalopathies. So the prion shape is the abnormal shape that is, causing all the problems. In the lab, we can actually make the protein form other shapes, but it doesn't seem to form them in nature, only in the lab. So they're not really relevant. If you have a bunch of this cellular PRP protein around and you introduce some of the prion PRP version, if you introduce like at least a few of them, then the cellular PRP is going to change its shape and refold to assume the prion PRP shape. And that actually doesn't happen with just one prion PRP protein. It takes a few. So if you just got one, it's okay. Then once you have these PRP-SC proteins, or the prion PRP proteins, they'll actually clump together. So the normal version of that protein doesn't form clumps, but the prion version does. And then those clumps just get bigger and bigger. The cell is going to keep making more of the cellular PRP because, it needs it for a job that's not getting done now, when all these PRP proteins are misfolding to form the prion shape and clumping together. All these new PRP proteins that are getting produced, they're misfolding as well as soon as they get close to the clump, and they're joining the clump, and it just gets bigger and bigger until it forms an aggregate that we call a plaque. We can also call it an amyloid, or we can call it an amyloid plaque. That plaque is itself going to keep growing, getting bigger and bigger until it's so big that the cell cannot survive. And, it's just going to kill that neuron that it's in, it'll kill neurons that are around it, and you're left with a little empty spot in the brain tissue. Over here you have a diagram showing that process. So here you have the nucleus with the PRP gene, or the gene for the PRP protein. That is being used to make more of these cellular PRPs, which have some type of function in the cell. They don't clump together. They remain kind of independent and go off to do whatever their function is. If you were to get some of the prion PRP proteins introduced into the cell, the cellular PRPs that were around it would actually misfold to form the prion shape. And then once you have multiple of those proteins with the prion shape, they clump together and form these little chains that just grow and grow and, get too big for the cell to contain and form an amyloid plaque. So you might have heard of the phrase amyloid plaque before, or at least amyloid, because that's also associated with other neurodegenerative diseases like Alzheimer's, prominently Alzheimer's, but also other ones like Parkinson's and Huntington's, also chronic traumatic encephalopathy, or CTE. CTE is the disease that young athletes can get if they have too many concussions as a result of the sport that they're playing. Anybody could get it, of course, if you have too many concussions, but that mostly just happens to athletes. Some of these people, when they die, if they happen to die young, if you autopsy the brain, you'll see that it looks like the brain of an old person who died of Alzheimer's, even though they're so young. So no one really knows what's going on in those diseases. No one knows what causes CTE or Alzheimer's or Parkinson's, except concussions for the CTE. But it's possible that prions might be playing a role just because the diseases also involve these amyloid plaques. So you're in these diseases, you are having like plaques that form and get bigger and bigger until the cells die around them. And eventually you've lost so many cells that you die. Or in the case of CTE, usually people don't die of the CTE, they die of something else, but it is affecting their brain and will theoretically kill them eventually. In the meantime, it does start kind of giving some of them cognitive changes or emotional changes, and affecting just, how they're kind of processing information, and their personality. So since these plaques are involved in all these diseases, there are some scientists that think that these are probably prion diseases, and that's an area of active research, although we don't have any conclusions yet. And finally, we'll talk about how those diseases are actually transmitted. So there's 3 different ways to get those transmissible spongiform encephalopathies. You could ingest the prion version of the PRP protein. You could inherit a mutated version of the PRP gene that kind of more commonly generates the prion form of that protein as opposed to the cellular form. And question mark. So sometimes, it's very rare, but sometimes somebody gets a transmissible spongiform encephalopathy and we just don't know where it came from or how they got it. And it seems like it just sprang up from nowhere. So I'm sure that it did not spring up from nowhere. We just don't know what the reason is why they have it. Out of all those 3 ways, ingestion is, of course, by far the most common way to get that type of disease. Any tissue of a person or an animal who is infected with that type of disease could give you the disease if you eat it, because that type of protein, the PRP protein, is found throughout the whole body. But the brain is where you have the highest concentrations of it, so if you're ingesting another part of the body, you might get lucky and not get the disease. If you're ingesting the brain, your chances are not so great to not get the disease because there's a lot of the prion protein there. So there's going to be a lot of, the prion form. If you're eating an animal that has, that like mad cow disease or something like that. In kind of a hospital setting or a healthcare setting, if you have a patient who has one of these encephalopathies, and their body fluids get onto some object, especially if it's a metal object, the prion proteins are going to stick onto that object. They stick really well onto metal especially. And then it's really hard to sterilize those objects in a way that gets rid of the prions. Then, if that object were to come into contact with a break in your skin or with a mucous membrane, like your mouth or your nose or your eyes, then those prions could be transferred to you, and at that point, you might have just caught that transmissible spongiform encephalopathy. So that's the reason why it's really important to destroy prions that are on objects that have come from some patient who has that type of disease and is being cared for. This is also one reason why it's really important to regulate the livestock industry, because what has been done in the past is that to feed livestock animals, sometimes you will actually take other livestock that got sick and died or old and died, dead animals, grind them up, and then feed them to the living ones. So if you have like a cow who has mad cow disease and has died of that disease, and then you grind that cow up and feed it to the other cows, now they all have mad cow disease. And then if you buy the steaks from those cows and eat it, now you have mad cow disease as well, very likely. So it's really important to kind of regulate what types of food can be fed to livestock, so that these spongiform encephalopathies are not just spreading throughout the livestock population and then into us. Actually, if you want to donate blood or donate organs, in order to determine your eligibility, one of the things that you will be asked is if you lived in Germany in the 1980s. The reason is because the livestock industry in Germany in the 1980s was maybe not so regulated, and there was some amount of mad cow floating around. And so if you lived in Germany in the 1980s, then you were eating in Germany in that time period. It's possible that you ate some prion proteins, in some of the meat that you had. and it's possible that you could have specifically mad cow disease. And that's why they will not let you donate blood or an organ or anything to anyone else and potentially transmit the mad cow disease to them.